can start it now. Yeah, let us start that now. So you got the protocol for amplification of ITS 1 and 4? You got the protocol. Okay, this group can start now PCR now. Okay. So who are all there in your group? Call the group members. Where is the DNA now? Who is handling? Okay, bring it here now. So all of you got your PCR protocols? Got it? So we need 4 protocols, one for amplification of 16S or DNA, another one is for ITS using ITS 1 and 4. Then we have 2 marker genes for Alstonia and 1 housekeeping genes for a gene for magnoporthic. You got everything? What is it? How many tubes are there? DNA?
So this is the DNA.
Okay, final room.
Yes.
Okay, good. So now you start your amplification now,
Yes,
right? Chemicals are with you.
sir, chemicals we ask for full sir.
Okay. So have you seen it now? It's completely dissolved.
Yes, sir. From yesterday itself we dissolve a -20.
Okay.
So it's in dissolve.
Right. Okay. That's great. Yeah. So these 2 DNA is from Bacillus.
Yes, sir. Bacillus.
Okay. So now these 2 are from the same sample, correct? It's only 2 replications. So you gave 0, 1 and 2. Now what you have to do is you can change the label now. Okay. Don't use the label 1 and 2. Right. You create a kind of code by which you can understand what you are handling. Because 1 and 2 is something which you cannot remember all the time, right? So it would be better if you use some kind of coding system so that you track the sample at any time, right? We have a coding system in our lab. We use the host or the source from where the sample was isolated, and if the identity is known partly, at least at genus level, you bring the genus name also there, and more importantly the place from where the sample is collected. So this is a triple code system. For example, if it is from rice, you put R. If the organism is Bacillus, you put B, right? Then you can put one hyphen, then the place you collected the sample, let us say it is from Delhi, or if it is from Almora, then you some coding system you evolve so that you don't forget it, right? So R, B, one hyphen, then put Almora, ALM. So you know it, the sample is from rice, and this organism is one of the species of Bacillus, and the whole sample was collected from a place by name Almora. So it altogether it is only around 5 to 6 letters, and if this kind of coding system is always needed, and you can track your sample at any time. You can bring some numbering system there, right? So that at least you know the samples are from one place, but 2 different samples. Some kind of coding system, so that you can track your samples at any time. This is very, very important. If you simply give 1 and 2, then this 1 and 2 never stays for long. So tomorrow you will be taking another 2 sample, and you will be giving again 1 and 2. So there is no end for that, right? So I am sure if you give a coding system like this, and the traceability of your sample is always there, that is more important, right? And again, one of the guiding principle here is whatever name you are giving, that remains throughout the life of that organism. much like the name which is given to you right from the birth till the end, right. So, what we do, the data generated from this sample also take the same name. If you are sequencing a particular gene from this bacillus and you want to give the name for the sample, the data is meant for uploading in some databases. This morning Dr. Anirban made highlights about a lot of databases from GenBank, EMBL, DDBJ. So, again the name goes with the data and the name goes with the culture also, you understand. So, here comes harmonizing the data sets and the culture with the same name. This is very, very important. Otherwise, many times you yourself will confuse what you handled a month ago. Those kind of confusions can be resolved. So, a proper coding system is very important. Of course, we know end of the day it is going to be identified up to species level, and you are going to give a species name for the sample what you are handling, if the purpose is to identify an organism. So, here comes the question, if two different bacillus species, let us say subtilis, if it comes in your collection, then you need to segregate for both of them. So, there comes the coding system here. So, this takes bacillus subtilis or B underscore ALM, right. Maybe another sample you collected from, let us say Srinagar, right. You give a different name. It may be R B underscore, maybe you give some name, maybe S N something Srinagar, so that you know this isolate is from a completely different location. So, here comes the coding system is having host name integrated and the geo name also integrated, and organism is abbreviated at the genus level. If the species is known, you can put the species also there. So, Bacillus subtilis is abbreviated as BS. So, RBS underscore ALM. So, this is a kind of internally made coding system. This is very, very important for microbiologists and plant pathologists. Tomorrow these names will take a central stage, you know, maybe a pathogen what Deepa is handling is one of the potential pathogen, extremely virulent, and people always call, okay, this isolate is very, very virulent. So, AN27 is one classical example. So, the naming is very important. AN27 is nothing but Aspergillus niger isolate 27, something like that, right. So, the naming is very important. Remember always in your mind, a small tube like this you cannot write too big, but you can easily write around 4 letters here or 5 letters here in the side. Okay. So, coding system is very important. Okay. Now, you proceed with the PCR Bacillus group, you are going to amplify 16S ribosomal DNA, and now you set up the PCR now. Okay. Anybody has done it, or you need a proper mentoring for that?
No, sir.
You will do it? Yeah. Where is the chemicals? Where are the chemicals? Yeah, fine. Any doubts on that, the coding system what we discussed now? So, I think the Kelex group is leading now, they are already with the DNA, and I quickly set up the PCR.
DNA to them another set
Okay, no problem.
to at least cope up that time,
Right.
because if every day is tracked and I don't know what quantity will be wasted. I think they are
Okay.
limiting.
So, any group already quantitated the sample DNA? Which group? Kelex group already quantitated. How much is the DNA yield? Can I have a look? Show to me. So, the group number 5, if I am correct? Group number 5. So, now they are leading the race now. They have A260 to 80 ratio. It's a very impressive 1.8. 84 is there. Okay, let us take it 1.84. That's pretty good in terms of DNA quality. So, and the DNA yield, they isolated 302 nanograms per microliter, that is 302 microgram per ml of DNA, and that is very good DNA yield for all standards. Another sample, I think they have isolated 184.6 nanograms per microliter. So, that is 184.6 microgram per ml. So, now what you need to do now, you need to make a template DNA. This is the stock DNA. Now, you make a template DNA which is having 100 nanograms per microliter. Okay. You just work it out. This is your stock, and you need to add, you need to mix X quantity of the DNA which is going to take out 200 nanograms, right, and you add the solvent whatever you used. So, you decide how much volume you want to make, right. So, let us go with around 50 microliter. So, you just calculate for 50 microliter of 100 nanograms per microliter, how much you need to take it from the stock. So, you simply put a small formula V1 N1 equal to V2 N2. You derive at the quantity what is needed from the stock and add the Y quantity of your solvent, you mix it well. So, if you are doubting your own calculations, your own handling, you can check it once again in nanospectrophotometer whether it is 100 nanograms per microliter or not. I am sure it will be 100 nanograms per microliter, plus minus around 5% variation may be there. So, that is expected also. Okay. Now, you go with 100 nanograms per microliter working stock. Just sit and calculate how much of stock you need to take, how much of buffer you will add. So, total becomes 50 microliter and the concentration becomes 100 nanograms per microliter. Do it now. So, that is true with other groups also. Okay. Any other group who reached this quantitation stage? Not yet. The kit based method you should be ready now. Okay. Okay. What about other group? Laboratory method? Okay. 10 minutes drying is enough, right? Now you add TE 10:1 now. Okay. Good. Yeah. You can make announcement. Tell them.
So tea is ready. So take your tea first, and then continue on.
Okay. So since the DNA is under drying mode now, you give a break, have tea, and come back.